me? Yeah. So thanks very much for that warm introduction, and it's really delightful to be here. I have many friends, old friends, as Lorna pointed out, that intersected with me during my scientific career. And interestingly, although I was at Stony Brook for 20 years, I never met Jim Simons. But our paths crossed without us seeing each other in many different ways. Of course, I knew about his legacy as chairman of the math department at Stony Brook. But I also lived a couple of blocks away from Avalon Park, which is a beautiful nature preserve that the Paul Simons Foundation established in honor of his son, who died tragically. And I used to walk my dog there every morning. And since he's not here, I can say he wasn't on a leash. But it's a really beautiful place, so his generosity has really touched my life in many different ways, and I'm disappointed I didn't get to meet him. Anyway, today I thought what I'd do is tell you about some of the approaches we're using and developing to study Rett syndrome, with the idea that maybe some of those will be transportable to autism and other neurological diseases. And so what I'm going to do in my talk today is give a brief introduction of Rett syndrome as a disease. And I think, as many of you know, it's due to sporadic mutations in a transcription factor called MECP2, or methyl CpG binding protein 2. And that acronym refers to the fact that MECP2 binds to methylated cytosines in cytosine guanine residues across the genome, and I'll be talking more about that later. I'm also going to talk about an unexpected role for glia that we found in this disease, which was considered a neuronal disease, and that used primarily a genetic approach. And then I'll talk a little bit about how we're beginning to get some preliminary insights into how astrocytes in particular may be interacting with neurons, and that's using a physiology approach in collaboration with a physiologist. And then I'm going to talk about a new method that we've been developing to help us try to figure out what MECP2 does, and to try to get at the gap between the loss of MECP2 and the aberrant behaviors that occur in this syndrome. And finally, I hope I have time to talk a little bit about whether I think Rett syndrome might be a good disease or amenable to the emerging gene therapy approaches. So Rett syndrome is primarily a disorder of girls, because it's on the X chromosome, and it affects about 1 in 10,000 live births of girls. It's characterized by a latent appearance of symptoms, like many other neurological disorders. Parents think they have a normal child for about a year, and then they start to notice that the girls are regressing. They have an overall retardation in growth, but their brains grow proportionately smaller, so they're slightly microcephalic. And that's due in part or associated with smaller neurons and more closely plaqued neurons, and also the neurons have less complex dendrites, so they're just overall smaller. The girls undergo progressive motor activity disturbances. They lose speech and purposeful hand motions, and they can acquire repetitive hand motions. And it's that constellation of factors which originally led Rett to be considered an autism spectrum disorder. But I'm not a physician, but if I see a girl with Rett and I see a girl with autism, they look very different to me, because the girls with Rett also have myriad physiological problems. And so Rett is now considered, I think, an autism-associated disorder, in part, I think, for that reason. The girls also have epilepsy and breathing abnormalities and a very abnormal gait. And they have a life expectancy of about 40 years, although some girls can live a normal lifetime, but if they do so with serious mental and physical handicaps. As I said, the disease is due to bad luck, mutations in the MECP2 gene. And interestingly, if you look at the catalog of missense mutations in patients and where they occur, oh yes, I have this 1960s pointer. So you have this beautiful screen, and then I'm surprised you didn't give me a pencil. So if you look at the distribution of where these mutations occur, you see that they're grouped in 2 particular places in the protein. And it turns out that those, of course, are the parts of the protein which are functional. So the cluster in the blue box corresponds to a portion of the protein that's been shown biochemically to be responsible for the binding of MECP2 to methylated DNA. And the cluster at the orange box seems to correlate very nicely with biochemical experiments that seem to show that MECP2 is a repressor, but I'm going to talk a little bit more about that later. I told you that MECP2 is born on the X chromosome. So because of dosage compensation in mammals, half the cells in a girl are mutant and the other half are wild type. It's not the case that they just have half as much MECP2 protein. And the mosaicism, I think, is shown very nicely in this slide. This takes advantage of a mouse in which GFP has been knocked into the endogenous MECP2 locus. So in a girl with 2 X chromosomes, if you inactivate the allele that doesn't have GFP, if you inactivate the allele that doesn't have GFP, you'll have a green cell. And if you inactivate the allele that has GFP, you'll have a black cell. So on the right, you can see you can see this by immunohistochemistry. DRAC5 is a marker for nuclei in red. You can see the cells that are expressing the modified MECP2 green locus, and you can see here in this portion of the hippocampus that about 50% of the cells are green and 50% of the cells are red. And this is something also that I'll be talking about a little bit later. And now boys can also have bad luck. They can, of course, get Rett syndrome as well, but because they're hemizygous, they have a more severe form of the disease, and so they die usually within the first 2 weeks of birth, and that's why it's sort of considered or discussed as a disease of girls. There are many mouse models now for Rett syndrome. The earliest ones were made by Cre-Lox technology, and they give rise to a completely null situation where MECP2 can't bind to, is basically completely non-functional. And the disease is less severe in mice than it is in humans. So for example, the male mice symptom onset is 4 weeks, and they usually perish after about 3 months. Both the male and the female mouse models are characterized by the same latency that you see in humans. It takes a while for the symptoms to develop. And then they have a constellation of motor defects which are similar in some sense or reminiscent in some sense of the patients. For example, the girls, I didn't mention this, but the girls have severe respiratory difficulties. They have apneas or breath holds and very irregular breathing, and it turns out the mice model this very nicely. In addition, the mice, as they become more and more or progressively symptomatic, they become hypoactive and sort of sit at their cage. They hind limb clasp, which is sort of a general phenotype of many neurological disorders or CNS problems. And also they mimic Rett in the sense that they have smaller neurons and reduced dendritic arbors on those neurons. So as I said, for many years, Rett was considered to be a disease strictly of neurons. And there were some pretty good reasons for thinking that. For example, deletion of MECP2 just from the brain using a brain-specific promoter in combination with Cre-Lox technology gave a phenotype that was similar to the germline knockout phenotype. Additionally, knocking MECP2 into a locus where MECP2 would be turned on in development just in neurons prevented some of the symptoms prevented some of the Rett-like symptoms in mouse. And finally, MECP2 was reported to be present at high levels in mature neurons and absent from astrocytes, which are the major, of course, glial cell type in the brain, and that was done by immunohistochemistry. Now, coming at this from a completely different perspective, as someone who didn't really set out to study Rett syndrome, and somebody who had, as Lorna mentioned, a long history in thinking about ubiquitous repressors, this last thing kind of struck a chord. It didn't really makes sense to me that a ubiquitous repressor would be excluded from non-neuronal cells in the brain. And I thought that was kind of an interesting point because if the disease was due to multiple cell types, that might explain why there were neurological symptoms, whereas even though MECP2 was absent from the rest of the body. And also the final reason for thinking more about this is just that I'm a curious person. And I was working with a postdoc who's also a very curious person. That's Rett Vallis, who's in the audience. And so together we decided we would revisit this issue of whether MECP2 loss was specific to neurons. And Rett was trained as a biochemist, and so we decided to take a biochemical approach. And the first thing we did was generate our own antibodies to MECP2. So one good thing about the Rett field is that people exchange reagents, and that's also one bad thing about the Rett field, because everybody uses the same reagents. So we made our own and showed that it was specific for MECP2 epitopes. And then Rett purified populations, different populations of glial cells and neurons, and then just did a Western blot and asked whether she could detect MECP2 epitopes in glia, just like in neurons, and the answer was yes. And then she did immunohistochemistry using a marker for specific glial types together with her antibody to MECP2, and she could see in tissue sections very clearly that glial nuclei contained MECP2, although it is true that astrocytes do express less MECP2 than neurons. So this set of experiments raised the question of whether loss of MECP2 from glia would have an effect and would contribute to symptoms in Rett. And a very talented student in the lab, Daniel Loy, took this on, again in collaboration with Rett. And so we decided to take a two-pronged strategy. We decided to take MECP2 away just from astrocytes and ask what the phenotypes were in the mouse. And we also wanted to do the converse approach, which was to add genetically MECP2 back just in astrocytes in an otherwise MECP2 null mouse, and then ask whether we would recapitulate Rett-like symptoms or to what extent we would recapitulate Rett-like symptoms. So our strategy was to use a Cre recombinase that was conditional, that was under control of an astrocytic promoter, which is a fragment of the human GFAP promoter. And we wanted it to be conditional because, of course, neural progenitors are also astrocytes. And if we removed MECP2 from the progenitors, then we would get both neurons and astrocytes that were lacking MECP2. So the idea was to remove MECP2 from astrocytes postnatally by treating the mice with tamoxifen. And when we did that, we found that the mice had a normal lifespan and they moved around the cage fine. It'd be hard to tell them apart from a wild-type mouse. However, they did have a pronounced abnormal respiratory rate, and the mice were smaller. So these results suggested to us that there was a role played by astrocytes, and so we continued on to the second approach, which was to do the Converse experiment and see whether we could rescue some of the symptoms. And the approach we used exploited a very nice experiment that was done by Adrian Bird's lab in 2007. And what Adrian's group did was create a new allele of MECP2 in which a stop sequence was placed upstream from the functional gene. And so the idea is that animals which bear this new allele lack MECP2 and therefore will begin to show symptoms of Rett. But then if you cross these mice into a mouse that have a ubiquitous promoter driving the Cre recombinase, you can treat with tamoxifen and remove the stop sequence and then restore MECP2 into the mouse. And if you use a ubiquitous promoter here, then you'll restore MECP2 everywhere in all the cells. And what Adrian's group found, strikingly, was that you could add back MECP2 to an otherwise null animal and rescue many of the Rett-like phenotypes, suggesting that at least in mice, the disease was reversible. So we exploited this approach, except instead of using a ubiquitous promoter to drive the recombinase, we used, again, the astrocytic GFAP promoter. And we were very kind of surprised to see that the rescue that we saw, this is now looking at a survival curve, using the astrocyte promoter was just as good as the rescue with the ubiquitous promoter. This was not a result that we expected, but it was quite striking. We then went on to look at other phenotypes to see what other aspects of the disease in mice might be reversible by just correcting the astrocytes. And we found that, as I showed you, the lifespan was significantly prolonged. The mice were nearly, but not quite, wild-type size. They had a very nice normal respiratory rate that was actually indistinguishable from a wild-type littermate. They had greatly improved locomotion and open field, moving around the cage. And the neurons and dendritic morphology were restored in regions where we had restored MECP2 to the astrocytes. So the first message that I want to relay is that our work suggests that Rett syndrome involves glia as well as neurons. And since we completed these studies, Narit Ballas and Yanni Kipnis have done similar studies looking at contributions of oligodendrocytes and microglia to Rett. And in both cases, there's an effect, although it's very different qualitatively than the effect of the astrocytes. We also showed that re-expression of MECP2 just in astrocytes rescues 4 of the key hallmark abnormalities in mouse models of Rett: premature lethality, breathing irregularities, hypoactivity, and neuronal morphology. So this then begs the question of what is the relationship between astrocytes and neurons? And it requires 2 things. It requires a better understanding of what's normally going on between astrocytes and neurons, and then experiments to test what might be wrong with that communication in the Rett model. And to address this issue, we turned to physiology. And the physiology was done by Daniel Leoy, who after leaving my lab as a student joined Paul Brem's lab to learn physiology. And so the first question was, what system do we use and what do we look for? Kind of a completely open question. So we decided to kind of focus on a paper that was done many years ago by Kang et al. In 1998, in which they showed that there was astrocyte-mediated potentiation of neuronal GABA signaling in slice. And what this group showed, so here we're looking at synaptic activity in the astrocyte and in a neuron below, and what they showed is if they stimulated the astrocyte, they could see a stimulation of frequency of events in the neuron. And furthermore, the effect of the astrocyte on the neuron appeared to be excitatory, even though the mediator was shown to be GABA. And that's shown by using the GABA-A antagonist bicuculline here, which shows the absence of the stimulated effect. Now, one problem with going directly to slice is that you don't know what circuitry is involved here. You don't know if it's a direct interaction between the astrocyte and the neuron. So we decided to take a step backwards and use a more simplified system to begin with. And so what Dan and Paul decided to do was to make a culture system in which you plate cortical neurons over a bed of astrocytes, and you plate the neurons at very low density. So usually neurons don't like to be plated at low density. That's something that Jerry knows very well from the old days. However, if they're on a bed of astrocytes, they seem to be a lot happier. And then the idea is to do a paired recording where we stimulate one cell and record from the other. So we can stimulate, for example, the astrocyte and record from the neuron, and then we can do vice versa. And I'm just going to show you the experiments where we stimulate the astrocyte. And that's shown in this slide here. So the top trace shows neuronal activity, spontaneous events that can be recorded. And then with astrocyte depolarization, this is electrical depolarization to mimic a train of action potentials, you can see that there's a pronounced effect on the coupled neuron. And what you see is the frequency, the amplitude is greatly enhanced. Now in the slide I showed you before, the effect was on frequency. But because we have such a large effect on amplitude, that's the predominant effect we have. We can't really look at frequency because it may be that we would just be missing, that the events are just bigger, so we'd be counting more of them. So we're focusing now on amplitude. And the quantitation of doing many experiments is shown on the bottom. You can see there's a flurry of activity of synaptic events after the depolarization, which actually outlasts after we finish the stimulus of the astrocyte, and then eventually goes back to normal. We then asked what the source of the currents was that we would be measuring. And so this is, again, is another representative trace from the neuron, and you can see that the activity is greatly reduced by the application of gabazine, which is another antagonist of GABA A receptors. And that quantitation is shown here. You can see that the event amplitude drops to 0 in the presence of gabazine. Dan and Paul also look at other parameters, one of which is shown here, which is the average holding current, which among other things reflects the number of open channels. And you can see that that also is dramatically decreased with the application of gabazine. Now, I'm not going to show you the data. This is spontaneous events, but actually the induced events by astrocytes on neurons are also gabazine responsive. So that means the combined data from the amplitude and the holding current suggests that there's a large tonic level of GABA, we think, being produced both by the astrocyte and the neuron. And then we found two kind of surprising things, which I'm going to show you. One is that we found that some of the astrocytes in the culture were capable of generating an action potential. And a representative one is shown here. And the events that we see in the neuron due to the astrocytes are sensitive to tetrodotoxin. It blocks everything, the spontaneous currents as well as the invoked synaptic events. And also, if we add the sodium channel blocker QX314 to the astrocytes, you see there's a significant decrease in the average event amplitude. It doesn't go to 0. And that's because although we're blocking the activity in the astrocyte, there's also synaptic activity coming from the neurons as well, which is probably due to ataptic synapses. Then we wondered whether the mechanism by which the astrocytes were exciting the neurons was through a conventional exocytotic mechanism. And this was the other surprise. So if we introduce BAPTA, which is a calcium channel buffer, into the astrocyte, you can see that the event amplitude in the neuron goes way down, consistent with a calcium-mediated event from the astrocyte affecting the neuron. We also uncaged calcium in the astrocytes, and that mechanism seems to be, we're bypassing the action potential here and just asking whether we can stimulate the neuron, and the answer to that was 0. So two kind of surprising things. One, that we can see action potentials generated by astrocytes, which suggests to me that under some conditions in vivo, the astrocytes are excitable. And second of all, that they're using a conventional exocytotic mechanism. And that's a little bit surprising too, because many in the field of gliobiology feel that astrocytes shouldn't be able to do that because they don't see by EM the same number of secretory vesicles that you see in a neuron. So I think that's an interesting question that we'll have to work on in the future. So now that we do have a robust response, we know what it is, we know how to elicit it, we know a little bit how it works, we know GABA's involved. We asked, is this mechanism any different if we use a Rett astrocyte compared to a wild-type astrocyte? So in that case, we make the same kind of cultures and do paired recordings. And in both cases, the recordings are between an astrocyte and a wild-type neuron. But in one case, we have a Rett astrocyte, and the other case, we have a wild-type astrocyte. And you can see very clearly, I think, that the Rett astrocytes are not nearly as capable of eliciting a response in the neuron as the wild-type. So the message from this group of experiments is that the synaptic currents that we see in our culture are primarily GABA-A mediated. Action potentials in both cell types look like they contribute to the synaptic currents. We think that calcium-dependent release in astrocytes is required, and Rett astrocytes have compromised GABA-A signaling. And of course, we're doing more experiments to further test these ideas, but it sort of it at least gives us a holding point to begin to understand how network effects might be affected by non-neuronal cells talking to neuronal cells. Now, I didn't tell you, but we also did experiments that checked whether the exocytotic machinery was intact in the astrocytes, and it looks like it is, which would place this defect upstream of the actual secretion of neurotransmitter. And so that leads to the question of the gap between what MECP2 is doing in the nucleus and these downstream effects. And one possibility is that the loss of MECP2 might be changing, maybe even subtly, the stoichiometric ratios of proteins in the cell. And so one way to sort of test this idea is to try a mass spectrometry approach to look at the proteome in a Rett cell versus a wild-type cell, be it an astrocyte or a neuron. And so Saurabh Garg, a postdoc in the lab, set out to examine the membrane proteome in Rett and wild-type mouse hippocampus. And the reason that we focused on the membrane to begin with is because whole cell mass spec analysis is a little difficult to do quantitatively because very abundant proteins like RNA proteins often get in the way. So we decided to focus just on the membrane proteins with the idea that maybe we'll see some differences in synaptic proteins, for example. And that would give us another clue about another way of closing that gap between what's happening in the nucleus and what's happening in the cell. And we used a quantitative mass spec approach, and which I won't go into the details, but I just show this piece of representative data where we identify the proteome from 3 individual hippocampi from 3 different mice, and that's shown here, wild type 1, 2, 3, and you can see that the results of the peptide counts are very, very close between these 3 different animals. And then on the right, you can see the mutant spectrum. And again, you can see they're very similar, but I'm sure it's caught your eye that the number of peptides is much less in the wild type than in the mutant than in the wild type for this particular protein, which is a mitochondrial protein. And in fact, I picked this representative protein for a reason, and that's because we got quite a surprise when we did the membrane proteome. So we identified 4,000 different proteins in the wild type and the mutant prep. And of those 4,000 proteins, and they represented many synaptic proteins and proteins in different intracellular membranes, of those 4,000, only 182 were changed between the wild type and the Rett using the threshold that we used, which was that we asked for 40% change, statistical change. And what was even more surprising to me was that most of the changes were down. Okay, that's okay, but they were mostly proteins dealing with mitochondrial functions, for example, electron transport chain and respiration. On the other hand, of the proteins that were upregulated, they also clustered too, but they turned out to represent RNA processing proteins, primarily RNA binding proteins. So I was surprised, but intrigued by the fact that so many mitochondrial proteins turned up until I went to the literature, always a bad thing. And I went way back in the literature, and I found something really interesting, which was way back in 1989, investigators really thought Rett syndrome was a mitochondrial disease. So this report did an EM study and reported that the fine structure of a mitochondrium was very different in a case of Rett than in wild type, and that was followed by a paper suggesting that Rett syndrome might in fact be a mitochondrial disease. And this was on human patients, and so on. So what happened, if you recreate history, is so interesting. So this group of papers noticed and studied in different ways metabolism and came up with the idea independently that mitochondria were affected in Rett. And then disaster happened, which is that the gene that is responsible for Rett was discovered. It kind of set the field back. Because then everybody focused on MECP2 because it's a transcription factor, and who cares about the mitochondria? What are the genes? What's going on with the genes? And so this sort of fell out of favor. But if you read the literature of neurological diseases now, it turns out that mitochondria and metabolism is having a renaissance. And it's possible, though this clearly is, the defects in mitochondria are not going to be a primary defect of MECP2. It may be an interesting pharmaceutical entry point to think about. So message 3, Rett syndrome pathophysiology likely involves metabolic deficiencies. We haven't tested that yet. Of course, we have to now measure oxygen consumption and do all the analyses you do to see what's wrong with the mitochondria and do our own EM. But if it turns out that there really is a metabolic deficiency in girls with Rett or in the mouse models, this could account for some of the overall growth retardation, smaller cell size, and even decreased synaptic activity. And this is quite interesting because a recent report suggests that the mitochondria at the synapse are actually different than the mitochondria in other parts of the cell. So I think that will be another aspect that we'll want to look at. So we're still looking at probably secondary effects, and we really want to close that gap between what MECP2 does and what the phenotype is in the mouse. And because MECP2 is a transcription factor, many of the experiments that have been done have been looking at the mRNA profiles in Rett cells versus control cells, with the idea that there should be large differences in expression of genes if you're missing this very important protein, especially if it's considered to be a repressor. It was considered to be a repressor because it binds to methylated cytosines in CG dinucleotides, and those are very much associated with gene silencing. So walks like a duck, talks like a duck should be a repressor. But the problem is when you do the mRNA profiles, what you find, so what one group finds is, well, everybody finds that there are small changes, so much less than twofold. One group reports that the changes are primarily up. So that would mean that MECP2 is a repressor because when you lose it, the genes are up. Okay. Another group reports that it's primarily an activator or a facilitator of transcription. So those genes that they report are mostly down. And then there's a group in the middle who says, no, wait, it does both. But that data is based on microarrays where you can't tell primary effects from secondary effects. So still crazy after all these years. So this is still something that has not been worked out. What we do, I think, all agree on is that MECP2 is primarily in the nucleus. If you look by histology or do cell fractionation, most MECP2 protein is found in the nucleus, like a very well-behaved transcription factor. I think we also all agree that MECP2 does bind to methylated CGs across the genome. In fact, that's how Adrian Bird first purified MECP2, was by binding to this epigenetic site. And recently, Adrian's group mapped all the MECP2 binding sites in vivo using chromatin immunoprecipitation. This is a snippet of that data where he mapped the binding sites of MECP2 along mouse chromosome 1. This happens to be from a glial cell, but it doesn't matter. It's the same from a neuron. He then mapped all of the sites of methyl CpG across the mouse genome. And then he aligned the peaks of where the methylated Cs were and the peaks for where MECP2 bound. And you can see from this little snippet, there's a very nice correlation. He then went on to do some calculations. There's a lot of molecules of MECP2 in the nucleus, about 10 million, which is much more than for a conventional transcription factor. This, which binds to a very small consensus sequence and occurs with a much lower probability across the genome because of that. So this amounts to about 1 in every 400 base pairs of DNA, which is enough to coat the genome. This being the case, we would have to say, I think, that MECP2 is not a conventional transcription factor that binds to specific genes through these small consensus elements. Rather, it binds globally at levels approaching a nucleosome, and I'll talk about that in a minute. And this, to me, to my lab, suggests that perhaps MECP2 is playing more of a structural role or an architectural role in the nucleus. So the nucleosome is the structure by which the genome is packaged into the cell. So you all know that the DNA is not naked DNA in the cell. It's associated with protein as chromatin, and also it's not linear. It's packaged into these repeated units called nucleosomes. And what they are about 150 base pairs of the double helix surrounding a core of proteins called histones. And of course, DNA is negatively charged because of the phosphate backbone. Histones are positively charged, so that electrostatic interaction helps tidy up this structure. Now, you can visualize these nucleosomes in the EM, and in fact, your textbooks, you probably all remember, talk about beads on a string. These are the nucleosome core particles that you see in the EM. Now, even though they're wrapped in these little structures, it's still possible, it's the genes that are associated with the nucleosomes and in between the nucleosomes are still accessible to transcription factors. So there's active transcription ongoing in these uncompact regions. In contrast, these nucleosomes can also assume a different conformation where they kind of wrap around each other and become even more compact. And when they do so, they form what's called heterochromatin, which you can also see in the EM as these big sort of amorphous black blobs. And the euchromatin in this picture are these little faint grainy gray lines there. Now, the heterochromatin is characterized by having methylated CG residues and also by the fact that the genes that are in heterochromatin are largely silenced. They're not accessible to transcription factor. The nucleosomes are quite compact. So when I say that perhaps MECP2 could be playing a structural role, what I'm thinking is that perhaps, for example, MECP2 could be involved in the pathway which separates expressed genes in euchromatin from silenced genes in heterochromatin. And if you mess that up, then you're going to have aberrant gene expression because genes are going to be in the wrong spatial location. And in fact, early work in Drosophila showed that if you artificially move a gene from the euchromatin into the heterochromatin, an actively transcribed gene, that gene will now be silenced just by virtue of being packed inside the heterochromatin. And then just one other thing I want to mention, which is the histones in the nucleosome also play a role. And that's because portions of the histone are accessible to post-translational modification, just like any other protein in a cell. And so this work began with David Alice many years ago and Michael Grunstein at UCLA with the idea that certain residues in the so-called tails of these histones, and there's 4 different types, can be modified, and depending on what that modification is, that may be associated either with gene silencing or gene activation. So for example, these flags that I have in green are modifications that are associated in yeast and in all kinds of mammalian cells with genes that are being actively transcribed. On the other hand, these red flags, these methylated residues, again have been examined from yeast to humans and been shown to be associated primarily with gene repression. Now, nothing in biology is all or none, but in general, that seems to be true. So if we want to test the idea that MECP2 might be involved in chromatin architecture, we need a technique which would allow us to look at heterochromatin, for example, and distinguish it from euchromatin. And we'd also like to have a technique where we can use antibodies to these different histone modifications and ask how they're spatially located with respect to heterochromatin, for example, in a wild-type cell versus a Rett cell. And I want to emphasize that this kind of analysis, this way of thinking about it, requires that we be able to look at individual cells. So if you do an RNA transcriptome, or if you do a Western blot, you are averaging the signal from all of the cells in that tissue. And I think what we really want to do is start looking at individual nuclei in the brain and ask what the effects are in spatial localization in the absence of MECP2. So we kind of scratched our head for a while to figure out how to do this. You can do EM, but then you lose out on looking at the antibody marks for the chromatin. You can do immunofluorescence, but the problem there is you want to look at a whole variety of marks changing with respect to heterochromatin, and the resolution in the microscopes for doing that with conventional microscopy, even with confocal, is not quite very good. So we kind of need a technique that allows us to do both. And a very talented postdoc in the lab, Mike Linhoff, decided to try to adapt a technique called array tomography, which was developed by Stephen Smith at Stanford, who's now at the Paul Allen Brain Institute, which was really designed to count synapses in the brain. And it was designed that way because his method allows really terrific axial resolution so that you can detect 2 proteins that are very close together as separate proteins. And we've worked very closely with Stephen to adapt his technique for counting synapses to looking at the nucleus. And the essential part of the technique is here. The part that's like EM is that you cut ultra-thin sections and you layer them on a slide. So these are sections that can be anywhere from 50 to 200 nanometers in thickness. And you plate them on a glass microscope slide, and they're embedded in an acrylic resin that Stephen's group developed, which allows you also to do immunofluorescence. And so you can take your slide and you can use any number of fluorescent stains. I'll show you we use a fluorescent DAPI, which intercalates into the brain and lets us see heterochromatin. We also use immunofluorescent antibodies to look at the histone marks. And then you image, and then you use software to reconstruct a 3D image from putting all your sections together. And after you've reconstructed and analyzed with one particular probe, you can start the process all over again. And you can actually do up to 50 different stainings for different markers, and then look at their relative orientation, either in a brain if you're interested in synapses, or in our case, in the nucleus. So now I want to remind you that the girls are mosaic and the mouse model is a mosaic. And what's really nice then for this technique is that Mike can draw a specific area that he wants to look at, and he can look at individual nuclei, and the wild-type nuclei, which you can see by taking advantage of the MECP2 GFP knock-in mouse, well, they're right next door to a mutant. So in the same section, we can look at a nucleus from a wild-type neuron and a Rett neuron and ask if their nuclei look any different in terms of their chromatin rearrangement. So I'm going to show you a movie which allows you to see what it looks like after we've done our 3D reconstruction. This is the CA1 pyramidal neuron from the hippocampus. These fluffy balls you see are heterochromatin that we've stained with DAPI. So they're the big silenced parts in the cell. So you'll see that. And then you're going to see that what the software does is it paints a structure that you want to quantify. So one thing I should mention is another really nice aspect of this technique is it's extremely quantitative. So we can get actual numbers for the intensities of whatever it is that we're looking at for our comparisons. So the software surrounds a surface for us that it's going to quantitate. Of course, we have to set a threshold. So in the experiments that I'm going to show you, I can't remember exactly what Mike's threshold is, but we're mainly looking at the larger heterochromatic foci, and we're not looking at the smaller ones. But we can set whatever threshold we want and do multiple exposures, depending on what we see. So you're going to see how the software paints that surface. And then I'm going to show you what an antibody to this particular epigenetic mark, methylation of K27, looks like. I picked this one to show you because it entirely coats the inactive X. That's known from many experiments before us. And you're going to see the inactive X pop up in this nucleus, and you're going to see that it's associated with the nuclear lamina, which is a silence domain. And that was sort of a confirmation for us that the technique was working. And then the whole nuclear surface is going to be painted so you can see how we define the volumes of nuclei. Now that I said this, probably won't work, but where's my, okay. So now it's going to color the surfaces that met the threshold that we wanted to quantify, and then you're going to see the inactive X. There it is, popped up against the nuclear lamina and surrounded by heterochromatin, and there's the software painting the surface for us to quantify. Now, if everything is working the way we think it's working, then then heterochromatin should be in a space in the nucleus that's not occupied where active transcription is occurring, which we can see by looking at an antibody for an activated form of RNA polymerase II. So this isn't going to be a movie, but this particular epigenetic mark should co-localize with DAPI. It's a silencing mark. And it should be reciprocal to the localization of where you see active Pol2. And I think you can see that very nicely here. At the threshold we set, you see the heterochromatic puncta identified by DAPI. They match this epigenetic mark, which is known to be associated with heterochromatin. You can see it's very nicely reciprocal to where the activated Pol2 is. And as I said, this is a very nice quantitative technique. And you can see from our data that as the heterochromatic density decreases, the amount of RNA polymerase increases. So it looks like our procedure is working pretty well. And I will show you another quick movie. Now we're beginning to look and compare a Rett nucleus with a wild-type nucleus, which is on the left. These are 3D reconstructions. And when I first looked at these pictures with Mike, I said, wow, I can see a difference. I think I can almost genotype because the MECP2 negative nuclei seem to have bigger and rounder heterochromatin than the wild type. And he said, well, let's quantify and see if you're right. Another good idea spoiled by data. No, actually it was right. So this is a plot of the percentage of DAPI in heterochromatin in wild type and mutant. And you can see that the mutant definitely does have a higher density of DAPI, which is highly significant. And it's not that the amount of DAPI is different in the wild type and the mutant cells. It's a redistribution, because if we actually calculate the total amount of DAPI, it's the same in both cells. And I was actually pretty amazed to see that comparing, this is composite data from different cells in different animals, and you can see how closely aligned they are in the amount of DAPI between the cells. And then we started looking at repressive marks besides trimethyl K9, which was known to be in heterochromatin. This particular mark is H4K20-methyl-3. This is another silencing mark, and you can see again that the density is higher in the mutant compared to the wild type, but the total amount is the same. So again, there's been a redistribution of that mark. But it's not that every mark is changed. This particular epigenetic mark, which is not so much associated with silencing, isn't changed between the mutant and the wild type. So there is some discrimination in terms of what chromatin is going where. And this is an interesting mark, this epigenetic mark, again, highly significantly enriched in the mutant compared to the wild type. This is a mark that's present in the gene body and is usually associated with genes that are being actively transcribed. However, recently, another idea is emerging, which is this mark can also be used to terminate transcription. So it's a little unclear what to think about the mark, but clearly it's enriched in the mutant in our case compared to wild type. And then an interesting experiment we did was to test the idea that was suggested by Rudolf Janisch in a recent paper in human iPSCs from Rett that were differentiated to neurons. His group claimed that overall transcription in Rett cells is reduced, and our data would seem to support that. So if we look at total amount of activated RNA polymerase II, that is the polymerase which is on genes that are being actively transcribed, it's an antibody that's raised against a specific modification in Pol2. You see that the mutants have significantly less activated Pol2 than the wild type. So it may be, in fact, that there's a global effect of transcription in the mutant. And then this was an interesting comparison where we plot total nuclear RNA polymerase II versus the percentage of this mark in heterochromatin. And what you can see is that this whole population of this mark is missing in the Rett nuclei. So that explains why, as I showed you before, there's more of this mark in the heterochromatin and Rett. But the interesting thing is, if you look at the amount of Pol2, you can see that the level is higher in this case compared to the global downregulation. So that would seem to suggest that there are cases in which, although the overall level of transcription is down, there are genes whose expression is more enriched in the Rett nucleus, which means that probably everybody's right, which is the best situation. And then finally, or second to last point, there was, using this mark, we saw something that we hadn't seen with any of the other repressive marks, in that there was a dramatic redistribution of the mark to the nuclear lamina, to these big repressive islands. So what the green and red refers to in this movie is that everything that's within a half a micron of the nuclear lamina is, what color is green, and then everything else beyond that is red. And so you can see that this mark forms these big islands in the Rett nuclei, which we've never seen in the wild-type nuclei. And then finally, it's been reported that the cells, I told you the cells, the Rett neurons in the girls and in the mouse models are smaller. It's also been reported that the nuclei may be smaller. And we see that there is a very slight reduction in nuclear volume in the Rett compared to the wild type. And you can, one thing I didn't point out, I guess I should point out two things. You can see that there's quite a distribution if you look at different individual nuclei. This is another reason why I think it's not so good always to average your signal. And the other interesting thing that I think is emerging from Mike's studies, which that's sort of why I put this slide, is he was looking at a population of cells in the CA1 region of the hippocampus where we would think that the cells side by side are all the same. But in fact, it looks like he's seeing two clear distributions, which is saying that even in this population of cells, which we might think is homogeneous, they're really not. There's some inbuilt heterogeneity, which I think is important to think about in terms of thinking about network activity. So message 4, we provide evidence that loss of MECP2 modifies 3D chromatin architecture. And we have data that suggests that MECP2 might be a facilitator of transcription because overall the level of transcription is down in the mutant, and there's a higher density of heterochromatin and repressive marks, and that would be most consistent with MECP2 acting as a facilitator. So when you take it away, you get more down regulation of genes. And our working hypothesis, and this is all I should have said at the beginning, this is very preliminary, but our working hypothesis is that maybe heterochromatin is like a sinkhole for DNA that represents a very low energy state for chromatin. And what MECP2 normally does is prevent euchromatin from coming into these heavily condensed areas and being repressed. And so we're continuing, obviously, to try to provide support for this. And then finally, MECP2 may be a repressor for some genes because we see higher levels of activated Pol2. So in the last couple of minutes, I'll just show some data which is already published, which suggests that Rett may be a disease which is amenable to gene therapy. And the approach that we took to test this, which was in collaboration with Brian Kaspar, was to use adeno-associated virus, which has been designed to incorporate any promoter that you wish to put in plus a transgene. And the nice thing about this virus is it requires a helper virus to replicate, so it doesn't replicate outside tissue culture cells where you can provide what it needs to replicate. The other nice feature is you can inoculate peripherally through the bloodstream. And once it's in the animal, there's no virus replication, and there's transgene expression. And of course, I just want to point out that the virus has to go through the blood-brain barrier. The brain is surrounded by blood vessels, and then there's a lot of mishigas that it has to go through to get from the surface of the membrane into the nucleus. It has to duplicate its DNA, and only then can it express the transgene, but it seems to be able to do that. And so we tested the idea, as I said, with Brian Kaspar, Sarab, and Dan did this. We injected both males and females with an AAV that carries MECP2. We did brain immunohistochemistry to make sure we were getting some expression in the brain. We didn't get great transduction, I would say. The highest level we saw was 30%. And the other kind of surprise was that depending on the brain region, we either got really primarily astrocytic expression or primarily neuronal expression, and then some regions were kind of equivalent. Nonetheless, we proceeded, and I'm just going to show you a movie of 2 male mice. One was injected with wild-type virus, another was injected with the MECP2 virus. This is the hypoactive mouse, male mouse that received the control virus, and there's the mouse that received the MECP2 virus. This mouse will eventually die here, but he lives for a significantly longer time without any symptoms. We also looked at females, and they did quite well as well. So this is a female mice injected with a control virus. These are female mice injected with the MECP2 virus. This is a sort of a general phenotypic scoring system that includes a lot of different phenotypes. Here's the wild type, and you can see that although the females that have the control virus keep progressing with the disease, the ones that received the MECP2 virus were stabilized and seemed to live a similar lifetime. So I think in principle, Rett syndrome may be amenable if we can base anything on the mouse studies. But we need more efficient and immunosafe vectors if we're going to proceed with this. And overexpression of MECP2 could be problematic because there's a duplication syndrome, but I can talk to anybody who's interested. I'm not so sure how much of a problem that's going to be. And I'll just end by saying whether I think anything we're doing on Rett has anything to do with autism. So clearly, Rett is a transcription factor disease, and that brings with it a very specialized set of problems that we have to deal with. However, I think there are some approaches which are applicable to thinking about autism. And one is approaches that look at cell-cell interactions between neurons and non-neuronal cells. I think it's very likely that these other neurological diseases are not just a problem of a neuron that isn't functioning well, but rather a problem of communication between different cell types. And it's fine to have the circuitry, and it's fine to have invisible brains where you can see all the neurons, but I think that secretion of neuropeptides and cell interactions are going to be something that we're going to have to focus on. And then another thing is that neurons kind of do fundamental things. They send out processes, and they make spines. And it may be that many of these neurological diseases all funnel into a common problem, which could be, for example, metabolism. So I think more and more evidence is emerging that many of these neurological disorders have mitochondrial defects. And so I think it might be worth thinking about whether we should look at that a little bit as maybe a therapeutic entry point for diseases other than Rett. And then finally, GABA. So I think that there's more evidence emerging that GABA imbalance is responsible for some of the network problems in autism and other disorders. And the finding that GABA can be both excitatory and inhibitory, depending on the context, adds another layer of complexity, but continues to shine a spotlight on GABA for thinking about network problems and other diseases. Thanks for your attention.